I would like to introduce Professor Girkin at Durham University. Thank you, Professor.
Thank you. Do I have to finish at 3 o'clock, or do I get till 3 p.m? Thank you. It's all right. Right. I'm glad you let me back after yesterday. What I'm going to do now is, I hope, going to answer 3 questions that people have asked me during intervals, both at lunchtime and yesterday afternoon as well. So it's taking what I did before and trying to push it a bit further and beginning to set things in a very clinical setting, particularly focusing on trying to do in vivo imaging. So it's going to concentrate, there'll be some optical sectioning, as you'll see in a moment, and then nonlinear imaging. And I hope by the end to talk about some adaptive optics and why this may be a very good way forward to start applying these things potentially towards the clinic. So the first of all, you'll see in a moment some of the problems and complications of in vivo imaging, these nonlinear methods, and then what we have to do to image at real depth. This is a slide I used yesterday, and you'll see later why I've put it up. But remember what we've got when we're doing, if you like, the confocal and normal fluorescence imaging. We're generating fluorescence everywhere, and then we throw a lot of this away and just let through the light that's generated right at the focus. This works. I mean, it's a very, very standard techniques. We can get high quality images. It's relatively cheap. In other words, a single investigator can buy a confocal microscope. We've now got advanced lasers. It's simple to operate. And it's a very useful way then with a computer to build up 3-dimensional images. So you sort of go, well, that's perfect. Why, what's wrong? Well, for a start, we're illuminating, as I showed in that previous slide, a large volume of tissue. And we're only taking the light from a very small volume. So we're wasting a lot of light. As you get scattering, the photons bounce all around the situation, and they blur out. We start losing contrast. And as we've heard yesterday, blue and UV light is not good, and everyone knows about this. It can be toxic. We also get the excited light being scattered, and generally this is a very, very approximate thing. But very often, if you want to image to 30 or 50 microns, confocal imaging works. Beyond that, depending on the tissue exactly, it becomes more of an issue. I wanted to touch on just for a moment something that I think and I know is becoming increasingly important in life science research labs. And some of you may or may not be aware of this. And this is zebrafish. So they are little fish. They have in Britain or in Europe, they have in tropical fish tanks. They are transparent, except for some stripes on them. They develop very rapidly. They are very easy to manipulate. So we can put green fluorescent protein and other things in them easily. But the most amazing thing is that for a very simple fish, they are a very good human model. So, for example, although the heart in a zebrafish, as you'll see later, only has 2 chambers, and your hearts all have 4 chambers, 22 drugs that work on your heart out of 24 work in exactly the same way on a zebrafish. If I do this in a mouse model, only 12 of them work in the same way. And a mouse model has 4 chambers. They're actually a very good model for the development of cataracts in the eye as well, which I can go into in a different thing. And just last night, I very briefly looked up to see whether they were being used for cancer. And there's 2 very high-profile papers that have a very large number of citations that indicate that the zebrafish is potentially a really good model, both for looking for drugs, but also modeling actually what's taking place in cancerous systems. So it looks as though they're also a very good model to be used in cancer studies. But we want to take images of them. We actually, in the case of what I'm doing, we want to take images of live beating zebrafish hearts. And you would want to do this perhaps in any organ in a zebrafish, but because there are problems with it. If we use confocal microscopy, the GFP and the other fluorophores, in the case of a zebrafish, bleach. Because what we want to do is image them for very long periods of time. In the research we're doing, we want to image them for 24 or 48 hours. In other words, as the problem develops. You can't do that in a confocal system. So we've been using this SPIM, which is single plane illumination microscopy. And so what we do is, if we have our sample, this is what it sort of looks like. If we look through a normal microscope, it's a mess. So what we do is we put a sheet of light in from the side. So let's say this is my zebrafish, and I put a sheet of light, a flat sheet of light in from this direction, and then I look here. So all I am illuminating is a single optical section there. I put a flat sheet of light, and I'm looking along this axis. So as I move my sample through, I can illuminate each individual layer, one at a time. So this means, again, I can build up a three-dimensional image. I'm only the plane that I'm illuminating, the section I'm illuminating, I'm using all the light from that area, and other areas are not getting any light, so there's reduced photo bleaching. And you can actually do this at very, very high speeds, because it's just however long it takes you to take a picture. You don't have to scan a beam around. This is the way that we set it up. So we have some optics, which produces a sheet of light. Here is the sample, and we take the image, and some of it goes onto a camera, which looks in fluorescence, and we also put some infrared light in from this direction, which goes onto a camera. So this one is just producing an overall image, in this case of the heart, and you'll see why we want that in a moment. So this is the scientific image that we want, and this is an image that we're going to use for something else. And this is what it looks like. So here you can just about see is the sheet of light going across here. Our zebrafish is in this tube, a pipette mounted vertically, and we anesthetize the zebrafish, and then the imaging takes place along this line along here. And we can keep the zebrafish, they're quite happy, they develop quite normally we've done it for over 72 hours held while we've been doing, not in that case imaging all the time. So, what are the advantages? We're only illuminating a single plane. That reduces the photo bleaching and all the nasty effects associated with that. The one complication is we do need a transparent sample because we're putting light in from the side. So it will be very difficult to use this on my finger, for example, because the sheet of light will just not go in, and more importantly, I won't get any light out through the front either. But I said I wanted to image a live beating zebrafish heart. And that's a bit of a problem. Some other people have tried to image beating zebrafish heart or developing hearts, and what they do, they put a drug in the water which stops the heart, then they take the pictures, and then they start the heart again. Now, if that was me, I wouldn't be very happy, and I think my heart wouldn't be very happy. The other way they do it is they take a very long video frame, a stream of video, and they go, well, it's nearly the same position there and there and there. It's not accurate enough. So what we've done is we've synchronized the imaging to the beating heart. So I said we take an infrared image, and I'm using this as an example of how you can pull together series of methods, put them all together to solve a really challenging biological or potentially clinical imaging problem. So we take a series of images from that infrared image. So we have an infrared image, and we record them at around about 100 frames a second. It's not very fast, and we just use a very cheap camera. So we record those images, but we do some image analysis on it, and then we can then trigger the camera, and in fact the laser that puts the sheet of light in, so just before the heart is going to be in the same position, we put the sheet of light in and take the picture. So we can do this by doing the image analysis and some special dedicated electronics. You could do this with an electrocardiogram by putting a wire inside the heart, but when the heart's only got about 200 cells in it and it's beating, it's not easy to do that. So what we do is we analyze the images, and we do it very simply. We just compare what changes on each pixel one frame to the next. It's called SAD. Perhaps that says something about which is sum of absolute differences. We just look at how the image is changing over time. And these green crosses is our SAD value as the heart goes through its cycle. So these images are just taken relatively rapidly through the heart cycle. And then what we do is, this is where the crosses are the SAD values, and we then join them up with a very, very simple algorithm, just looking at 3 points to find, so this point at the bottom there is where, in this case, we want to take our image. So what we do is, just before the heart's going to be in the right place, we tell the system, fire the laser, so the electronics then, by the time it's taken the electrical signals to get through, fires the laser and takes the image at exactly the same point in the heart cycle. So what you'll see here, if I use the mouse, is this is the beating heart. It is slowed down, and you can see the blood going through, and you can see the heart wall moving. And what you've got here is a very boring image, because the heart wall is not moving, and these are images taken with the heart in the same position each time. All you see are the different blood cells moving through. So we've now, in fact, you can do a detailed analysis of this, and we can capture the position of the heart to within about 1 micron on each occasion. So what we can now do is we take a continuous image, predict when it's going to be in the right place, fire the fluorescence laser, the blue laser, and then we move on to the next section. So we take an image, and then we can slowly, or not slowly, we can step through the heart. And this is what you then get. So again, what you'll see here is the beating heart, and then this is an optical section at one point, and you'll see the heart keeps on beating, and slowly the optical section changes as we move and take deeper and deeper images. So hopefully that will go on. So you can see that this is about 200 microns right across here. So these are optical sections taken from that beating heart. So we can now take these optical sections and begin to build them and put them together into a three-dimensional process. Now, I think what's going to happen in a moment, I had these structures, so we've actually got some better images than this, which people, if they're interested, can see later. But we can actually fluorescently label it and build up a three-dimensional image. I use this is the postdoctoral researcher who did this, put this together, and he gave me the image, and I said, it's much better if you can make the image rock. Is this going to work? If it doesn't, do it this way. And this is what happens when you give a video to somebody. It stopped moving. It should be. Oh, wait a minute. It should. Oh, I'll give up. Shut it up. What? I don't know why. The video should be rotating. It's probably because I'm trying to play it off the memory stick. Let's just go back one. And then go down. And now that's better. I'm not sure whether this makes it any better. But you can see now that we've got the video rocking the heart backwards and forwards. And we can therefore now look, and we can actually take this one stage further. So this is reconstructing it with the heart in one point in its cycle. We can then clearly do that and take exactly the same image, but at a number of different points in the heart cycle. So this is now an image that's built up. So these are the various, there's a valve at the back of here, you'll see. So this is now a three-dimensional image that you'll see rotate, but the heart is actually beating as we do this. So that's the heart beating, so this is taken from a live zebrafish. This takes about 5 minutes to take. I forgot to loop it. I'll give it one more. Is it going to go again? So this is taken from a live, and we can do this now with about 2 micron resolution. So this has been reconstructed to smooth it all out, but we can actually watch and track where each individual cell moves within the heart. So this has given us the ability to build up and take these four-dimensional images. And as I said, this is in a zebrafish, which seems to be becoming a very, very standard model now in a number of different biological areas. But I now want to move on slightly from that. So overall, we said yesterday that the challenges are in optical imaging. We want high sensitivity, high spatial resolution, high time resolution, and minimally perturb the sample. And we've said, okay, with the SPIM imaging, we've minimally perturbed it, but we're still putting some blue light into it. So the other technique, as was mentioned yesterday in answer to one of the questions, is this technique of multiphoton excitation, where what we're typically doing in the case of fluorescence is we have a fluorophore that, let's say, needs blue light to excite it. So we put 2 photons of roughly twice the wavelength, half the energy, into the sample, and that they can be absorbed simultaneously, and they then produce blue fluorescence, or the longer wavelength fluorescence normally coming out at the other thing. But the chance of this happening, of infrared light producing blue fluorescence, is very, very low. And it only happens right at the focus. So now we have a method, if we take we take infrared light, shine it onto our sample, only at the focus will we have enough light to generate fluorescence. And you might just about see, so this is a sample, it's actually of safranine, and there is a green laser coming in here generating the sort of that cone of light you saw. And those of you with sharp eyes may notice that when I turn the pointer off there is a little yellow spot just hanging in space. And that's where 2 photons have been absorbed simultaneously to produce the fluorescence. So it's just an example. If anyone's interested, this one picture was taken by Brad Amos. Is a sort of classic, is the classic picture of this method of imaging. And if you I'll show it to you afterwards if you're interested. But we can generate it. The only trouble is this doesn't happen very often, and we need about 1 kilowatt of laser power to do this. Now, anyone that, if you do photodynamic therapy, knows that if you put 1 kilowatt of laser power on something, the therapy part it's basically smoke you get. So how are we going to try an image using 1 kilowatt? Well, the way we do it, or the way this is normally done, is we use a laser, a femtosecond laser with a very, very short pulse. So typically, so the average power, the bit that does the heating, is probably about 1 milliwatt, but the peak power is 1 kilowatt. And if you want to know what this means, so we've got a train of very, very short pulses coming out, about 80 million a second typically. And to give you an idea of how short the pulse is, in 1 second light goes round the Earth 7 times. Our pulses are about 100 femtoseconds, and they just about cross a human hair. So it gives you an idea of the speed. So very short, very intense pulses. So the poor tissue, it excites up the fluorescence, and it then thinks, oh, there's a lot of laser power, I ought to start heating up. But by the time it's thought about it, the pulse is finished. So the thermal effect is, for all intensive purposes, negligible. The way we do the scanning is typically we take the laser in, we bounce it off 2 mirrors, we focus on the sample. And the other thing is, because the fluorescence has only come from 1 small spot, we don't need a confocal pinhole. Every fluorescent photon is a good photon. We can collect them all. So we can put detectors in transmission, or we don't have to go back and de-scan the light. We can collect the light directly at the back of the microscope objective. Or we can go through, scan it, and bring it back there. So we can collect these photons, because the crucial bit now is we're only exciting them in a very small volume. And these are, there are hundreds of images. This is a living blood vessel that was built up. This is a very, very thin sample, but you can see this is DAPI staining the nuclei, so you can build up 3-dimensional images. You'll see some later on as well. So what's the take-home message? We're putting in longer infrared light, which means the light is not being scattered as much. Long red light doesn't get scattered, as we've heard a lot about that. There's also a big difference between our exciting wavelength and our emission. Exciting's in the infrared, our detection is down in the visible. Because it's infrared light, generally it's less phototoxic, we can collect all the light, and we're not going to get any bleaching. We could get it in the focal plane, but outside that, there's no excitation. It's infrared light. We don't get anything. And because we're using very, very short pulses of light, we can also do fluorescence lifetime measurements. So can we use this in something? So we built it up. So we were working with somebody who was very interested in doing slow and controlled drug release. In this case, this is a silicon rubber implant, and this is clearly just a blue dye to demonstrate. But they want to sort of put these, and in this case, they particularly wanted to do it for delivering drug through the white, through the sclera of the eye. And this is what it looks like in a dynamic state. So this is using this is actually loaded with rhodamine 6G again. The drug we used in the end was naturally fluorescent. But this is where the silicon rubber is, and what you will see, just using a normal fluorescence microscope, is this burst of dye, could be drug, being emitted. And this is then a plot in the corner here of the intensity of the fluorescence with distance going out, and the colors are different time points. So we can obviously do this in water, but we wanted to do this in an eye. And so this is the way we do it. This is a living eye. It's a freshly excised eye. This is a bullseye, and we can intubate it and basically keep it alive for about 24 hours. You can keep the vital functions taking place. And what we do is we place one of these implants just on the sclera, just on the downside towards the cornea. We look with the microscope uphill. So in other words, what we're expecting from what we knew is that we'd expect the drug to move round towards the back of the retina through the sclera. So we're actually seeing the drug, if we see it, going uphill. So we're not giving it an advantage of letting it roll downhill. And this is what we see as the image to begin with at different depths. This is after 5 minutes. We see very little. And then after, 80 minutes, we can now see the drug beginning to follow along the collagen structure. So this is now down at a depth of 500 microns. So using this multiphoton technique, we're imaging, in fact, we can go deeper than this, right the way through, and we can track where the drug goes, and we can therefore modify, or the chemists could modify the silicon implant to adjust the rate at which the drug got delivered. This would not be possible using confocal microscopy because the light just gets scattered too far. The problem is, though, I said in that case, the drug we were looking at was naturally fluorescent. In the other images that I showed you, we'd labeled the sample with DAPI. But is there a method that we could actually perhaps not use a fluorophore? Because we do get this bleaching problem and we can get phototoxicity. And so one other method that can be done, again, is a non-linear method, is so-called second harmonic imaging. So this is where we put light of one wavelength, let's say it was in the infrared, and let's say it was at 1064 nanometers. And then in front of it, we have the right kind of molecule. So in this case, an asymmetric molecule. What you will get out of it under the right conditions is exactly half the wavelength. So it's not being excited up, it's a non-linear process. Again, if anyone's interested in the physics and the maths, I will talk to them about that afterwards. The crucial bit is there's no fluorophore. It's because of the shape of the molecule and the way that the light wave drives it vibrationally, or not quite vibrationally, the way it drives the electrons around. And the wavelength comes out at exactly half. And this green laser pointer works on second harmonic generation. So inside here is a laser that's operating at 1064 nanometers. There's then a crystal at the front, and the light going onto that then comes out at exactly half the wavelength, twice the frequency, i.e., in this case, green. It's very good. The light is only emitted, or primarily emitted, in the forward direction. So it doesn't come out in all directions, so that could be a problem. It's also very good. I said it has to be an asymmetrically shaped molecule. It also works at interfaces, at cell, for example, under the right conditions and cell membranes. The other bit that's important is that certain types of collagen have the right requirements for this to happen. And I always forget which ones it is, but if I remember correctly, it's type 1 and type 3. But if anyone comes back and says it needs to be type 4 and type 2, but I think it's 1 and 3. So you can then image using this. So we don't need a fluorophore, but we do need the right shaped molecules. And this is now a method of combining the two things. So in here, the blue parts are from collagen or from muscle fibers, and the yellow parts, in this part when we look over here, the blue parts, the collagens and muscle fibers, and then the yellow parts here are pericytes, a sort of type of stem cell that are expressing yellow fluorescent protein. So we can begin to put these methods together in combination. But we still need a molecule in there that's got certain properties. So can we get away without having that? What we would like is something that's chemically specific, but where we haven't had to add anything extra to it. And so the technique that was developed initially by Sunny Zhu at Harvard is CARS, Coherent Anti-Stokes Raman Scattering. So what happens here is, the downside is we do need 2 lasers now, and you'll see why we need them. So what happens is we have 2, in this case initially, 2 energy levels, and we put light at 1 wavelength in, which is called the pump wavelength. And then we put in a second laser at what's called the Stokes wavelength, which actually, if you like, drives the molecules down an energy level. So they don't go back right down here. We've driven them down, or in fact what we've driven is vibrations down to that point there. And then normally what happens is another photon is absorbed, which is the same as the pump wavelength, where it then gets excited up again. And then in the end, we detect this wavelength that's coming from this higher level. So this is going to be the shortest wavelength. So these 2 wavelengths are the same. They're probably in the infrared. So is this one. And then this wavelength comes out at the end, and that's the one we detect. And again, now, this signal is going to depend on the intensity of that, the intensity there, and the intensity there. So it depends on the intensity of those 2 squared and that one linearly. So again, we're going to get localized things. Another way of looking at it is this way. So if these are my 2 wavelengths of light going in, the pump wavelength and the Stoke wavelengths, they're going at slightly different wavelengths, different frequencies. So they go together, and when you have 2 waves of different frequencies, if they're coherent, they beat together. So we then get this slow vibration, and what effectively is happening is that vibration is causing the molecules, certain molecular structure in it, to vibrate. So we've now got, and then from that vibration, we can then see some emission. So we've now got a method of doing this, and the way we do it, again, you'll see is very standard. We get the laser light going in, and I've lost where it's coming in. Here we go. The laser light's coming in through here. It goes through some scanning galvos onto the sample, and then in this case, we can either collect the light in the forward direction or in the backward direction. And again, we don't have to actually add a confocal pinhole, but this is done on a microscope that very often has a confocal pinhole in it, though you just tend to open it right up. So this is the method. It's exactly the same as the other one. We put the laser in, we beam scan across the sample and collect the light in the forward or in the backward direction. And you can look at some beads, and you can measure the resolution, and you can then take images. So these are all images, and the contrast mechanism, the thing we're looking at, is the vibrational modes that are present. So these are all taken in 2 seconds. These are ones that Sunny gave me a couple of years ago. And so this is looking at fat, because fat works particularly well here. This is stratum corneum. So what you're looking for is samples, molecules that have a good Raman, a good vibrational spectra, and then you adjust your lasers at the right wavelengths to get that beat frequency that then drives the molecules. You can actually be specific. You can label things slightly. So, for example, if you replace the hydrogen with deuterium, the frequency of the vibration is slightly different. We've added some weight to it, so it's going to vibrate at a different frequency. So this is, in this particular case, in some saturated fatty acids, you can actually deliberately put in some acids that have been deuterated, and they have a slightly different vibrational frequency, and therefore you can look at you can do an image at one frequency, i.e. where the lasers are tuned to one vibration, one that's at another, and then slowly you can watch this over time. So for example, you may have put in an external thing to see how it's diffusing in. to see some dynamics that are taking place. And then you would do a standard method of overlaying the various images. In this case, it's been overlaid with some fluorescence as well. But you can look down here to see how things change. So you can slightly label things by using deuterated materials. So why might you want to use it? Generally, there's no labeling. There's no photo bleaching. We've got no fluorophore in there. You've got contrast due to the vibrations and due to the chemical things, and you can get 3D optical sectioning because it's non-linear. It depends on the intensity of the light raised to powers. So CARS is, again, another very powerful method. You have to be a bit careful because you will excite all the particular vibrational bands, so it's not going to be the same as if you use a green fluorescent protein, which you know is only being produced by one particular bit of the cell. This would excite everything that had that molecule anywhere in the cell or anywhere in the sample would be excited. But there is a method, therefore, of imaging in this manner. Downside, you do need 2 ultra-short pulse lasers or a tunable laser that you can get 2 wavelengths out of. So it's becoming more complicated. And if you remember back to one of the first slides I showed yesterday, you're only going to bring out these sort of big guns to do the imaging when you have to. In other words, when you want to go deeply, when you don't want to put fluorophores and things in there. Choose the right tool for the right job. Don't do this for doing a really simple fluorescence-based assay. Say, oh, well, I don't need to put the fluorophore in single cells. Yeah, but if you can do it cheaply, do it simply. So a quick summary. We've looked at multiphoton. So if you have multiphoton, generally speaking, it's going to be with a fluorescence material in it. In this case, for the multiphoton fluorescence, you require a fluorophore. Yes, it works very well for GFP and YFP and all the other colors. And you can do time-resolved measurements. So these techniques that you may have heard of called FRET, where you have 2 molecules and the energy gets transferred from one to the other. The fluorescence lifetime changes, so it works very nicely for FRET. You can do harmonic imaging. 2nd harmonic or 3rd harmonic imaging works very nicely at interfaces. You need the correct shaped molecules or structures. And then you can do CARS. You don't need any labeling, but it's not going to be specific. It is specific for a particular chemical, but not how that chemical or where that chemical has been produced. Any chemical that has that vibrational mode will be excited. So it's chemically specific, but not where that chemical may have come from. But taking it a little bit further, I said that what I wanted to do with this, and generally what we want to do, is we want to go as deeply as possible. And my reasoning behind doing this, obviously, if you wanted to do any of these things in the clinic, you can't kill the patient and you don't want to take optical sections, so you want to go deeply inside to see what's going on. If you're trying to do life science research, you want to follow things in vivo over extended periods of time. Because a lot of what happens in looking at things that happen over time is, even now, what you do is you have a series of animal models. And it's like looking, for example, at trying to work out the rules of a game of football, or soccer if you're American. And you go to a football match after 1 minute, and you dig a hole in the middle of the pitch, and you point your camera in one direction, and you take a picture. And then you go to another match after 2 minutes. They're playing the same games. So you go to another match after 2 minutes. You dig a hole in what you think is the same place in the pitch, and you point your camera in what you think is the same direction, and you take another picture. And you do this 90 times, and you work out the rules of a game of football. It would be really difficult to do. That's what happens in many animal studies at the moment. You keep having to make time sacrifices because you perhaps can't do the in-depth in vivo imaging that you want to do. So, but one of the problems, if you want to go in depth, let's say you want to look several hundred microns in, and it's very difficult. As we go deeper in, now I will accept that Philadelphia cream cheese is not a biological sample. In fact, it's an entirely man-made material, right? It has no cheese in it whatsoever. It's just fat and water and protein stirred together. But these are images that were taken. So at the top, this is 10 microns in. You can just about make out that they're little balls of fat. In fact, what they are, they're about 2 or 3 micron balls of fat covered with protein in water. And the protein sticks the fat and the water together. But as we go deeper in, the image is getting more and more blurred because each one of these little fat droplets is acting like a lens, and it's steering the light around. So it comes back to the thing I was saying yesterday about aberrations. So now, instead of my light here going nicely in and nicely back out again, it's all being steered and moved around. It's not being scattered. That would mean it bounces. It's actually being refracted. It's being bent round by the fat or the differences in refractive indices. And I use this as a gross example. So this means by the time we get deep inside a sample, our images are all being aberrated. They're being distorted. So we're losing resolution, quite clearly. So what can we do about this? Clearly in a piece of tissue, we're having to go through the cell membranes and there might be lipid deposits in the middle and this sort of thing. So that's the equivalent. Well, it's the same problem that astronomers have. So astronomers want to look at the stars up in the sky, and they want to do it from ground-based telescopes. But they have to look through the atmosphere. And what the atmosphere does is it distorts the image. So what they developed were what are called adaptive optics. Optics they can change the shape of very rapidly, so they can correct for the aberration. So if they know that the wavefront is bent in one direction, they can put the opposite shape on the mirror and correct for it. The way they do this in astronomy is they often shine a 50-watt laser up into the sky, so we might have to change it again for doing our samples. And this is an image, although I now work in an astronomy department, this is the Google Galaxy for adaptive optics. I have no idea where NGC for, someone said it's the spiral, I really don't know. But you can see here, this is what it looks like before the adaptive optics. So this is the atmosphere blurring them all out. Now we can begin to see the individual stars or the individual things that are present. So can we do this in a microscope? So if we think about it, in a perfect situation, this is a simplified way of thinking about it. If we have a nice, perfectly flat wavefront going through our sample, going through our microscope, through our sample, we will get that perfectly focused spot. But if we now put a sample in the way here, what's happened now is our wavefront has got distorted because the sample has optical properties. Bits travel faster or slower in different areas. So my spot has now got bigger. It may not even be round. So what can we do about it? So if we had some light that was coming in here, so this is for multiphoton imaging, if you like, and we want to make that spot as small as possible because that's what sets our resolution. If we give the incoming beam the opposite shape, so we have some optical element which bends the light this way, when it then hits the sample, which is bending it the other way, what we can end up back with is a nice flat wave. So in other words, what we're doing is putting on the opposite shape. We can do exactly the same thing. We've done it in our SPIM microscope, where we don't alter the light coming in, we just look at the light coming back out because we know it's being distorted. So we can do this, we can apply the opposite shape on our optical element to correct for the aberrations that are present. So the way that we do this, for people who are interested, we have a mirror. I've just realized don't go on. No, I apologize. When I've put this together, I've taken out the one slide with the optical diagram in it. I apologize profusely for that. So basically, in the case of the multiphoton or the CARS microscope, there is a picture in a moment. Let me just go on for a second. So the way that we do it in a beam scanning system, this is in CARS, we have our light from the laser sources, so this is 2 lasers if you remember for CARS, goes in, it goes through a beam splitter onto our mirror that we can change the shape of, a deformable membrane mirror. The light comes back, it's now got a shape, it now gets reflected because we've changed the polarization, goes into the scan head, gets scanned on the sample, and then we build up our image in the normal way. And what we will actually do then is, what shape do we need to put on the mirror? So if I go back through here, we can actually calibrate the mirror. And the way we work out what shape to put on is we choose some metric, some value in our image. So it could be contrast, it could be brightness. There's a number of different things, and different methods work well in different situations. And then, or we could actually talk about, measure what the error is in a set called Zernike terms. So I said yesterday we can get spherical aberration and different types of aberration. Mathematically, these get called Zernike things. So we could deliberately put a certain amount, and let's choose the simplest aberration, defocus, certain amount of plus defocus, certain amount of minus defocus, and have a flat mirror. And from those three images, we can then begin to join up the dots and calculate how much aberration correction we need to put on. We actually do this for 20 Zernike terms and keep correcting and adding more and more correction. But our metric is saying, is the image getting better or worse as we add this correction on? And this is what it looks like for a bead. And we can see here, it's very interesting. I'm running this off the PC here, and obviously when I goes from a Mac to a PC, that should say 1 micron bead. So this is a 1 micron bead that's obviously horribly distorted because of the sample that we're looking through. By the time we've added the corrections, and this is the value, this is our metric value, I think this was measuring intensity. As we go up here, you can see that our metric value is getting better. It keeps going up and down as we put on plus and minus terms. And in the end, we can get back and significantly improve the image. As I said, we can do this in a cars. This is adaptive optics. We can either put the the mirror here, or we can actually put it after the adaptive. And this is the sort of image that you get. So this is purely looking, because it was what was available in the local, when we did this about 5 years ago now, in a cars microscope. This was available from the local butchers just down the road from Sunny Zoo's lab. We had a problem with the person writing the paper here, who actually wanted to say, you should always say where you get all your materials from, and he actually wanted to give the name of the butchers that we'd bought the bit of chicken from. It didn't go in the final version of the paper. This is what we get. So this is looking at a CH stretch. It's a particular band. And this is what it looks like with no correction. This is what it looks like. Now, in this case, we're not correcting for the aberrations brought about by the tissue, but purely due to the aberrations in the optics of the system. So before I started doing this, I thought I could set up optics. I've learned I'm rubbish at it, because you set it up and you think, oh, this is the best I can do. But actually then, when you let the system correct for the aberrations that are just present in the system, you get a much better image. And if we then, in this case, correct for the sample aberrations, it gets even better. In this case, at only 260 microns in, we got a 10 times increase in signal and a 3 times increase in resolution. And we can do this, and we can actually now, there are methods of doing this fairly rapidly and in real time. Now is not the time, this is not a specialist talk on just doing this. But there are ways now of correcting for these aberrations. In this case, we were getting, in the end, more signal 750 microns into the sample than they previously got at about 50 microns. And we had to stop at 750 because the objective had hit the tissue. So you can really improve the signal. That means if you improve the signal, you can use less laser power, which means you might be able to use it perhaps more in the clinic. This is an example. This is actually using it in our SPIM system. And the image on the left is the uncorrected one. This is the image on the right. And as I play it through, you'll be able to see, I'll just choose somewhere at random, how much clearer the image is over here. In fact, you can see very clearly, and if you look at it manually, individual cell membranes, whereas here it's obviously just become a complete blurred mess. So there's probably a route, and I'm sure that this got touched on earlier, that you can look towards true in vivo imaging, where you can use micro-optics, and that was touched on in the just before with some of the things that are being done, for example, very definitely at Rice, but also at Stanford and other places. You can integrate the adaptive optics and make it dedicated. You can use optical fibers, in this case a special one called a photonic crystal fiber for collecting the light. You've also seen that there are ways that we can use imaging to compensate for movement or effectively optically freeze the motion, and you can begin to think about handheld microendoscopes. In fact, this is moving it towards medicine. So these are some images that are taken from a group up in Edinburgh. So this is a normal histological image. This is looking at it when you start to add in the cars and the second harmonic imaging that they were looking in here. And there's a number of people who've been looking at this. It's beginning to move towards the clinic. In fact, the one I know of is Carsten Koenig's group, who are based in, help me back, somewhere in Germany, and I've forgotten exactly. They have built a clinical instrument that is being used for skin, colon, and breast cancer diagnosis. So this is using multiphoton, so it uses fluorescence, and it also uses harmonic imaging for building up and doing clinical imaging in practice. So this is taking the techniques that I've talked about here. They are going and beginning to move into the clinic. Probably, for most applications, it's overkill. It's probably more than you really need if you can do it more simply. But there may be some examples. For example, during someone who is having breast cancer surgery, at the moment, very often they have to take the bit of the diseased tissue out, they take it down to the pathology lab, they freeze it, they try and do it, and then they bring the results back. Well, with this, you perhaps could move your scan head, you've perhaps had to open the breast up, or perhaps you can just do it with a miniature probe and get some signals straight back from it. You've not perhaps had to add any exogenous fluorophores. So there are ways that you can then do. And crucially, this is looking down, remember, at the cellular level. There's then a question of when is a cell actually cancerous or not, but that becomes a slightly different question. This was touched on, and I want people here to think about this. And I'm going to come back to it whenever I'm speaking next week. We have now developed a miniature microscope. It has no scanning optics. It's basically a wide field microscope. It doesn't do any optical sectioning. And roughly, it hasn't quite come out. It's 2 millimeters along here, and it's 3 millimeters down here. And it basically is made and machined of a single piece of plastic. You can do this using diamond turning, so it's possible to machine optical quality, and you can actually make these surfaces any shape you like, which is an important thing here. So it's not molded, it's been specifically machined with very high precision. This actually, the way it was shown here, has an optical fiber, but we've actually got one where we literally put a miniature LED directly on the back of it. And you can now buy commercial cameras it's just the chip that is 2 millimeters or 1 millimeter square with 756 by 756 pixels. And you can take that down at the bottom and put all this in a small package. You do need an external battery. We don't have a battery. So we can make a microscope. It can work in reflection or fluorescence. We can't alter it, it's fixed. It's fixed and designed to do one particular job. But you can specify what that job is. So it could either be focused right in contact, or we can adjust the focus to be 0.5 millimeter in. It's not confocal, so there would be blurring, but you may want to look through a membrane and look deeper inside, kidney being one example. It can image at up to 30 frames a second. There's obviously got to be enough light to do it at 30 frames a second. So there is a limitation. And basically, there's a small battery on the back which then broadcasts the signal wireless. I can't give you more details than this because at the moment it's going through a patenting process. I've not given away any secrets in what I've done here. But we can make cameras this size. And what I'm going to do, and I will come back to this, so the people that are in the audience here that are going to be around next week have an inbuilt advantage. I'm going to be horrible. You're sitting there. You're sitting back passively. In a moment, you're going to ask lots of really good and important questions. At the next talk, I want you to talk to me. I want you to be giving me the ideas for what we can do with that instrument. But I don't just want to say, oh, I would stick it here, I would put it here. That's easy. I want you to begin together, and I will give you time in the lecture for the people that aren't here as well, to begin to think, to talk amongst yourselves. How might you think about using a camera, a microscope that's that size? But not just thinking, oh, I would put it here. What might you have to do? So, for example, if you were going to put it inside a mouse model, what genetic manipulation what's the disease, what's the process you want to look at? Actually begin to think about designing an experiment and putting all the bits in here. You've got a collection of people here who are chemists, who are physicists, who are biologists, who are clinicians. You should be able to think together, to put them together. Now you've got a wonderful time. You don't want to do the dancing this evening. Dancing's bad for you. It's all right, that's a personal comment. Or tomorrow, sorry. So, and you don't want to spend all your time drinking beer. Well, actually, drinking beer and doing the thinking is a really good way of doing it. Just give it a little bit of thought. You've got plenty of time. You'll meet people. You're all in the same hotels. Just begin to talk amongst yourself. You have a microscope that's 3 millimeters. Draw it out. Think how big it is. 3 millimeter cube. What might you want to do with it? And what other bits of science do you want to bring towards it? And then we will review this whenever it is. I think it's the 16th next week. Hopefully, I've given you an idea. So in the case of nonlinear microscopy, it's been around 21, 22 years. And it's actually a really interesting example of how multidisciplinary science takes place. So you need very often optical physicists, laser physicists, engineers to put all the bits together. At the commercial level, special lasers, femtosecond lasers were around, but they've now been specially engineered just for use in biology. And it's used more and more for real biology. When you're thinking about methods, select the right tool for the right job. Use these complicated systems when you want to go deep and in vivo. And when you want to go deeper, you need to use adaptive optics. Not a subject here. You may need to even change the shape, the time shape of your pulse. Miniature options are available. And I've only sort of brushed the surface. And as I said in the first talk, and I'm saying it again to you now, go away, go and think, go and talk to each other. There's some opportunities here. Again, I put those up just so that people are aware. And again, I thank you very much for your attention.
Congratulations for the nice talk. I have one question. When we have multiphoton events, we have low probability to happen the events. So you take great power and pulses. But then you have probability to have saturation of single state excitation and high order photo damage. There has any solution to that?
No, I mean, you're still, if you're using a fluorophore in there, you're still going to have exactly the same problems that you have with using normal one photon fluorescence, if that's sort of so, and in fact, to be honest, sometimes it can be worse in multiphoton excitation, because what can happen is, for example, sometimes it's good, 3 photons get excited and you get ultraviolet, but the other thing that can happen is your fluorophore, let's take fluorescein, for example. If you get 3 photons into fluorescein, very often that can be quite short wavelength UV, and then the molecule falls apart. So we still have, if there are photochemical effects taking place, they're still going to take place even in the multiphoton thing. The good news is they're only going to take place at the focus. So that's the good news, yeah, but in the focal plane, it can actually be worse. There are some papers that were published by Dave Piston of Vanderbilt University about probably 10 years ago now, who actually showed that actually in the focal plane it can be worse. But generally the good news is it's only going to happen in the focal plane, and if you're doing an XYZ section, you quickly move out of it. I would make one other comment, that there are, for those of you that would be aware of the quantum mechanics that's taking place, we're actually exciting up slightly different energy levels by 2 photon excitation compared to 1 photon. There is now some work being done, and I think there is a real advantage and big advantages that can be done where people are trying to produce fluorophores which are specifically excited by 2 photon. Different, again, as I said, that the structure needs to be slightly different. There's somebody in Durham that's making some compounds, they're called retinoids, and they actually affect stem cells. They affect a lot of things, actually. But they are being tailored specifically as well to have a large 2 photon cross-section. So there is some real advantage that can be done. So instead of using the standard fluorophores that are lying around, ideally, all our chemist friends should be making these wonderful fluorophores that are optimized for 2 photon, and the rules are slightly different. Hope that helps.
So thank you for the lecture. It was good for me. I have a couple of questions, but the most interesting for me now is how can you do the correction of the wavelength shape to how can you do this?
How can I correct the aberrations?
How can you correct? Oh, so and the next is how can you obtain 10 times more signal with this correction?
Well, so if we think about it, that was 10 times more signal. So as the light, in the case of the multiphoton, as the light goes in, instead of being focused to a nice small spot, the spot has got bigger. So as the light's gone in, instead of going straight in and being focused, it's now hit a ball of fat. So instead of coming to a nice focus, it comes to a bigger focus. It still gets focused, but it's bigger. The effects that we're looking at, so my resolution is set by the size of my focal spot. So if my focus spot has got bigger, my resolution has gone down because my light is not being focused to such a small spot. The other thing is, if my spot gets bigger, my light is spread over a larger volume. So I've got the same number of photons, but they're now in a bigger volume, bigger area. And that means this is a non-linear effect, and it depends on the intensity of the light, which is the number, if you like, the number of photons divided by the volume. So if my volume's gone bigger, my intensity has gone down, so my signal falls. So if my light, let's say, was going in and then got distorted, so let's say now that I bring the light in at a slightly different angle, which is one way of looking at it, it then goes through, it doesn't get distorted in the same way. I've now got my small spot, my resolution has gone up, and my signal has gone up because I've now got all my photons coming from this smaller volume. And it depends on the square. And that's roughly why, yes, you'll see, I don't want to get into it, it doesn't completely depend on it's a little bit more complicated than that. But yes, that's roughly why the resolution went by about 3 times and the other bit went by 10 times. That example there is cars, and you've got three laser pulses being involved, so it's a little bit more complicated. But generally, that's what happens. And also, the aberration changes the shape in that direction compared to that direction. Because remember, the resolution, the size of the spot, depends on the numerical aperture in the plane and the numerical aperture squared in the other one. How does the mirror change the shape? So the mirrors that we use, it's a very, very thin membrane. These are a very, very thin membrane, and it's 1 micron thick, and it has a metallic coating on one side. And behind it, there is an array of electrodes. They don't touch the mirror, but there's an array. And then when you apply a voltage to it, it pulls the mirror in that direction. So you arrange the ones, the images there were either taken by one that had 37 actuators or 52 actuators. So they're made by either a company called Optical. So what happens is we can then pull the actuators and change the shape.
You can control the tilting.
Yes,
by the voltage.
Well, you can do tilt if you want to, but tilt is actually a bad one. We correct any problems with tilt on a different. A tilt is a waste of our adaptive optic mirror, because you can do tilt very easily just with a piezo-controlled mirror mount. So tilt and what's called piston, we don't deal with this way. It's the other aberrations that we deal with.
Very good. Thank you.
mean, if anyone wants to go, yes, please ask a question. Sorry, I was trying. So if anyone wants any more details, I'm very happy to go through it. The aim of this was to give very much an overview, as everyone has been doing, and then ask us specific questions. Yes, sorry.
Okay, so it's about the details of the small objective or microscope that you showed at the end.
Yeah.
What are the specs on it? What's the NA coming out of the front or the magnification?
What would you like it to be? That's what you've actually asked. Exactly the first question. Sorry, it's the second question.
Okay, at least 0.55 NA on
that's
the front,
possible.
and at
so
least
there is
2 to 3
so
magnification.
there is a trade-off. So it's going to depend on what working distance you like, and there are. So we know that we can make. So we have made, not with the complete microscope, but we've made a single lens. It uses a lens and a mirror. And we can get a numerical aperture of 0.8 with a working distance, I'm going now from memory, of about 200 microns.
In air?
That is in... Now you're asking, I'm trying to remember. That was designed to work in air, yes. But the other thing is, because of the way it works, if you want to do it in water, we design it to work in water. So that's why... This is brilliant. You've asked, so these are, if you're trying to, I've given you, said that I can make a microscope that does these things. That's the sort of question. So what magnification do I need? What resolution? And if you change the resolution, there's a trade-off with working distance. So roughly, to get the 0.5 millimeter, I think the NA was 0.2.
I mean, do you have any, I guess what I should have asked is, do you have any hard cutoff limitations for that size and material that you're using?
Yes, but off the top of my head, I can't
Okay.
remember what they are.
Okay.
So yes, there are. So we can change the plastic that they're made of slightly. So we have some other gains. The other part that, if we go into the details, if you wanted it to work in the UV, which we could do, the parameters change slightly because the plastics have quite a large change in refractive index with wavelength. So we can correct for it, but there are sets of parameters. So you're right. I've left a little bit.
No, let's thank Professor.